This will be our discussion of cell biology, and that's going to include certain processes that take place inside the cell, like the precursors leading up to protein synthesis, as well as cellular replication, otherwise known as mitosis. For this section, it's going to be important to have a basic working knowledge of the structures that we see intracellularly. Now, we're not going to be getting into each of these structures in depth, but as we talk about the cell and we talk about different tissues, it's going to be important to at least know what the terms inside the cell are. We'll have to understand the membrane more than any other part of the cell. That outer covering on a cell is going to be the factor that determines how and how quickly different molecules and ions get into and out of the cell. So that's going to be the basis for when we talk about membrane transport in our next section. We're going to look at the stages leading up to protein synthesis. Now, we already talked about protein structure, amino acids linked together in a long primary sequence. This looks at how do we determine what that particular sequence is? Where is that information stored in the cell? And then finally, mitosis and DNA replication. So when we look at the cell, I want us to first realize that this is a human anatomy and physiology class. So without having to mention this every time we say the cell, for our discussion by default, we are going to be talking about eukaryotic cells. We're not going to be talking about the prokaryotic cells or cells that might be found in plants, algae, fungus. We're talking about a stereotypical eukaryotic human cell and its relevant structures. This is a diagram that you probably would have seen something similar somewhere along the way. And I'm sure at this point, studying the cell isn't new. You've been introduced to this before. The first thing we want to point out is the inside of the cell is going to have a number of different organelles suspended in a fluid-like component called the cytosol. These organelles, plus the cytosol, collectively inside the cell is called the cytoplasm. So when it comes down to, we mentioned the term membrane, plasma membrane earlier. Really, a cell is the cytoplasm contained by the plasma membrane. And when we look at that cytoplasm a little closer, it's the cytosol with the various organelles suspended in it. Any of the fluid found inside the cell, and for all intents and purposes, the fluid inside the cell is the cytosol, can also be referred to as intracellular fluid. Likewise, anything outside the cell is going to be termed extracellular fluid. So again, when we look at our next section, kind of looking ahead to set that up, and we talk about items getting moved into and out of the cell, we're moving those particular molecules from the intracellular fluid into the extracellular fluid and vice versa. Of course, there are other fluid components. We have the plasma that makes up the blood. But for now, we're going to keep our discussion focused on the cell, the cytosol, or the intracellular fluid, versus its immediate extracellular fluid, which can also be termed interstitial fluid. When we look at the other items that may be inside the cell but don't quite fit under the heading of organelle, we can get to peroxisomes. This is not going to be considered an organelle structure. It's a chemical entity. It's going to reside in the cytosol, and it's going to help maintain chemical homeostasis inside the cell. So it's going to act as a detoxifying agent, an antioxidant agent. It can act as a catalyst for different chemical reactions taking place inside the cell. Another structure that's going to be important is the cytoskeleton. Again, not really considered an organelle in the true sense of that word, but definitely an intracellular structure. For certain cells that we'll talk about this semester, the cell's shape is going to be critical. Some cells really want to maintain a sense of three-dimensional shape and structure in order to carry out its function. Other cells are going to be very flexible. One of the first cells you'll talk about in AMP2 is going to be the red blood cell. Very flexible membranes. We can take this red blood cell and shove it through the narrow openings of the capillaries as we go from artery, capillary, and vein. But picture the cytoskeleton as a tubular network inside the cell to give that cell some sort of rigid support. When we do integument, you'll see the cytoskeleton become more practical. There's one layer in the skin that we call the stratum spinosum. The cell has shrunken in, and it looks like it has spicules sticking out. Simply, that's the membrane has shrunken, and now you're seeing that internal structure become more obvious as literally the skin of the cell kind of shrinks around the skeleton. Now, as far as organelles go, we're looking at structures that become analogous to organs in the human body. Right? We haven't delved much into the human body yet, even though it's an A&P class, but we're getting there quickly. When we think about the organs that you already probably know about with their general function, what do the kidneys do? They filter the blood. What does the stomach do? It's digestion. The lungs for gas exchange. Every organ has got this basic tagline of function that can be attributed to it. 5 words or less, you can slap a function on that organ. And all of those organs together carry out very specific roles. They work together to maintain the viability of the organism. In the cell, similar sort of analogy. We've got certain organelles, they all carry out certain aspects that help that cell maintain viability. We've got some organelles that are more with the metabolic symptoms. We've got some that are associated for growth and transport within the cell. So as we go through the different organelles, we kind of list them off. I'd like you to get a nice tagline for each of those organelles. Again, we don't need to go into them in great depth, but you should know basically what they do. You can slap a function on to each of those in 5 words or less, you'll be home free. The first one is the lysosomes. The lysosomes are going to be the digestive organelles, if you will. They're going to be vesicles inside the cell containing digestive enzymes. If that cell takes in another entity, like a bacteria, the vesicles containing these enzymes can merge with that was just uptaken, and use the enzymes inside to help break it down into smaller subunits. The mitochondria, you've all heard the tagline for this one, powerhouse of the cell, pretty much in every introductory biology book you'll ever find. The mitochondria is called the powerhouse of the cell because that's where we see the majority of ATP get produced. And we've already been through this. For every glucose we break down, we get an average of 36 ATPs. The overwhelming majority of that came from activities inside the mitochondria with our Krebs cycle, our beta oxidation, and our oxidative phosphorylation. So the mitochondria is going to be known to produce ATP. Now, we see DNA contents in mitochondria. And at one point, my slide hasn't been completely updated, it was mostly maternal DNA. Now we know that there's probably more paternal DNA as part of the mitochondria than we once thought. Next up is our endoplasmic reticulum. Now this is going to be considered a transport system. It's a multi-lobe structure that spans inside the cell, and we can use that structure to transport items inside the cell from one place to another. This becomes relevant for what we're going to talk about today, leading up to protein synthesis. We'll see the code, if you will, the plan for making any individual protein is going to be stored in that cell's DNA. We need to have a way to recruit and transport a copy of that code, as well as transport the building blocks that code is going to need to make a specific protein. we're going to rely on the endoplasmic reticulum for that. To tie that in just a little bit closer, we have two types of ER. We have rough ER and we have smooth ER. To continue on with that lead up to protein synthesis, we get to the ribosomes. This is where protein synthesis occurs. That's what this organelle does. It takes the code from the DNA, copy of it. It takes the amino acids that it has on hand, and it simply binds them together to form the protein we need. Now, how this ties into the ER is that the ribosomes are a small organelle that sits on the surface of the endoplasmic reticulum. If we see endoplasmic reticulum with ribosomes embedded on the surface, it looks like it has a rough surface, hence rough endoplasmic reticulum. If an endoplasmic reticulum portion lacks ribosomes embedded on it, it looks smooth, therefore that's going to be considered smooth endoplasmic reticulum. The Golgi apparatus. Sometimes you'll see this called Golgi complex. It's a packaging and sorting organelle. Again, closely associated with the endoplasmic reticulum. We transport within the cell, then we can package and save. We could package together waste products to be later on removed from the cell. So this is a packaging and sorting organelle. And then lastly, we get to the nucleus. Now, I know at one point, at least for myself, you always thought of the nucleus as like the most important organelle in there. It's like the brain of the cell. It doesn't really work like that. The nucleus is the place where we see genetic information stored. We'll see DNA stored in the nucleus. We'll see copies of that be made into RNA as we talk about the processes for protein synthesis. DNA, I want you to picture as two backbone strands, like the parallel horizontal portions of a ladder if it was leaning up against a wall. And connecting those two strands are these horizontal rungs. The horizontal rungs are made up of pairs of nucleotides. And in DNA, we've got guanine, cytosine, adenine, and thymine to make up the rungs of that ladder. On one side, you'll have guanine. Guanine always only ever binds to cytosine. So on one strand, you'll have a guanine. On the other strand, you'll have a cytosine. They'll bond together to form one rung of that ladder analogy. Adenine always binds to thymine. So you will see these GC and AT rungs of the ladder, just like we see on the diagram. Always, cytosines binding to guanines, adenines binding to thymines, never really getting away from that pattern intentionally. But you have two rungs of our ladder run parallel, horizontally, and then the stepping parts of that ladder. It's a double-stranded structure. Now, these two strands have a directionality to them. That's going to be important. We label one end of the strand, the 5' end, and we label the opposite end, the 3' end. But they run in opposition to each other. So you'll see on the left-hand strand of this diagram, the 3' end is at the bottom, running towards the 5' end at the top. The complementary strand, the 3' end is at the top, running to the 5' end at the bottom. Now, you may have heard the DNA structure get described as a double helix. The fact that we've got two strands gives us our double. We twist it to form a helix. So it's like we took our ladder structure and simply twisted it up to give that spiral helix shape. Now, in order for us to look at protein synthesis, the code for every individual protein is going to be stored as a gene on that DNA strand. We're going to go from DNA to final synthesized protein in two general steps. The first step is called transcription. That's where we make a temporary copy of our original DNA code. That temporary copy is called messenger RNA. I almost said mitochondrial RNA. Messenger RNA. So that copy of DNA is called messenger RNA. Messenger RNA. Think about that as it's the messenger. It's carrying the code from one place to another, just like a messenger would if they were delivering a note from one place to another. The messenger RNA is a copy of the DNA, and it carries that copy to the ribosome. And the ribosome will take that messenger RNA, read it, and through a process of translation, create the actual protein. So we looked at messenger RNA. You'll see a couple other types of RNA pop up, so I just want to throw some of those at you there for now. RNA as a whole is considered a single strand mirror image copy of what we would have perceived as DNA. That's what we picture when we think messenger RNA. The big difference structurally is that we don't have thymine in RNA. Instead of thymine, we have uracil. So we still have our guanine, cytosine, adenine, but instead of thymine, we have uracil. So whenever we would have saw an adenine on our DNA strand, we pair that up with a uracil while we're making our messenger RNA. The other types of RNA that you'll see pop up, number one is a ribosomal RNA. Picture that as part of the structure that helps carry out protein synthesis. It's an integral piece of the factory, if you will, piece of the ribosome. Transfer RNA, or tRNA, is going to be involved with handling the amino acids we need. Because remember, we're going to have this long strand messenger RNA, a copy of the gene from the DNA. We need to read that messenger RNA and associate parts of it with different amino acids. So we get to this one part of the RNA, and it calls for a certain amino acid. Well, we will need a transfer RNA bound to one of those amino acids to come and pair up. Then the next little section of messenger RNA calls for a different amino acid. Well, we will need a transfer RNA specific to that amino acid. So by running our way down that messenger RNA, its specific code attracts transfer RNAs with the specific amino acid that we're looking for to complete a particular primary sequence. Now, we look at this in a diagram. We're going to look at a video in just a moment. We look at this as a diagram, and we see our spiraled DNA. We're going to need an enzyme called RNA polymerase to untwist our DNA at a particular point, read that DNA strand. So it's going along here, and it's reading adenine, guanine, cytosine, thymine, cytosine, so on. It's reading it, and it's producing, as it goes, it's pairing up those nucleotide bases and lumping them together, linking them together to form our long strand, single strand, messenger RNA. So let's look at it in movement now. It's a dynamic process rather than looking at it as a strict, a still shot. To get an impression of what's going on here, let's look at it in motion. So here's our DNA. We have a particular spot on that DNA called the gene. This is where the information for a particular protein is going to be stored. Just before we get to that gene, we have a segment here called a promoter. It's going to attract our RNA polymerase enzyme. It unzips the DNA temporarily, and we're looking at the template strand. We only need to copy one half of this, remember? We don't need the other half. This is a single strand structure as far as messenger RNA is concerned. Now, our RNA polymerase runs along the section where the gene is. Now it undergoes a process called elongation. And that's where it starts reading the DNA and attaching the complementary pieces of RNA to make our messenger RNA strand. That messenger RNA strand now can go on to the ribosome. It's carrying a copied piece of the code from the original DNA. So we use the term messenger RNA as someone bringing a message from one place to another. You go to one place, you get the source material, you write it down, you leave the source material at the library, but you take the copy portion and you run off somewhere else where you can utilize it. So translation now, we take the messenger RNA. So we're at the ribosome now. That messenger RNA that we copied from the DNA in the nucleus makes its way to the ribosome. And in a very similar but opposite way, we read the messenger RNA and we look at the pattern of our nucleotide bases. Every 3 nucleotide bases is going to be code for a particular amino acid. So we can recruit a specific transfer RNA that has that amino acid we're looking for based on the 3-digit pairings of nucleotides. So we see the next one to get bound, there's an adenine, uracil, uracil. Knowing that adenines only ever bind to thymines and uracils are going to bind uracil is going to bind to adenines, this is going to be the code on the transfer RNA that would bind to those particular sets of nucleotides perfectly, bringing with it a specific amino acid. So this transfer RNA now would bond on, and what's happening just behind it is these 2 prior transfer RNAs are taking their amino acids, linking them together with a peptide bond, and all the while we see this growing segment of protein. They're developing the primary structure of that particular protein. So each transfer RNA has a 3-digit nucleotide code that will be used looking at the messenger RNA strand to determine which amino acid has to be bonded next. And again, just like we did with the transcription, now let's look at translation. So the messenger RNA leaves the nucleus and heads to the ribosome. The messenger RNA has all these nucleotide bases, and it's essentially the code for the entire protein, but divided up into 3 segments that we're going to call codons. Each 3 segment is going to associate with the 3 nucleotide portion of a transfer RNA, but each one carries specific amino acids: methionine, lysine, serine, glycine. And when you look at the potential combinations, the number of bases, 3 letters per codon, there's more than enough variability for every single amino acid. So for lysine, AAG, AAA, AAC, and AAU will all code for very specific amino acids. And by that way, we get our primary sequence of a protein. Transcription, going from DNA to RNA, and from RNA to protein is translation. Now, when we look at cellular division, 1 cell becoming 2, we have to start with an initial process that's somewhat similar to what we saw with transcription. Different process, but to visualize what's happening, understanding what happened in transcription helps you with cellular replication. This time, we're not going from DNA to RNA. This time, we're taking one DNA and copying the entire thing so we can have two exact copies of DNA. If we want to take one cell and split it in two, we need to have a copy of that genetic code for each of those new daughter cells. We had RNA polymerase as one of the main enzymes for copying and producing messenger RNA. This time we got DNA polymerase for copying the DNA. Spiral structure, the first thing we have to do, unwind and unzip. Unwind and unzip. Still remembering we've got two different strands, 3' to 5', 3' to 5' running in opposite directions. This is called the replication fork. DNA helicase binds at the junction of that replication fork. What you're going to notice with DNA helicase, as it unwinds, there's going to be something further upstream here that as you keep twisting it is going to build up tension. Topoisomerase is another enzyme that's going to bind up here, disconnect the DNA, let the tension relieve, and then reattach it. But the DNA helicase unwinds, unzips, and it's going to allow our DNA polymerase to run along each of those strands, making a copy. And what's going to be left at the end is one copy of DNA and another copy of DNA as we copy each individual strand. Now, if you look at this diagram closely, you will see that overall, yes, it's the same thing that gets accomplished, but they do it in slightly different ways. Remember, we've mentioned the orientation of each strand. 3' to 5'. We will always only ever copy DNA from its 3' end to its 5' end. So as we split, we get what's called a leading strand with a free 3' end, and we get a lagging strand, but this time we've got a free 5' end. And we only ever read DNA for copying it from its 3' end to its 5' end. So, piece of cake up here. The free 3' end, we start there. We run our DNA polymerase along there, and we simply match up the base pairs. If we get to a cytosine, we throw a guanine on there. We get to a thymine, we throw an adenine on there to partner up those base pairs. The lagging strand, though, awkward, we don't have a free 3' end. We can't read from this free end and work our way that way because that's the opposite way we would read the DNA. So what we do is we unzip far enough and we throw down an RNA primer at a certain point, and then we work from that primer backwards. So we're still moving into 3' to 5' direction along that DNA. So we lay this one down first, we put down another primer, we lay down another segment here, we lay down another primer, and we do another. We lay it down piecemeal in portions called Okazaki fragments. So continual copying here. Further we unzip, we just move that DNA polymerase along, copying as we go. On the lagging strand, when all is said and done, we've got these individual Okazaki fragments now. We have to go back in and remove these RNA primers and kind of fill in the gaps again. So again, let's look at this happening in animation form. Helicase unwinds, unzips. Creating our replication fork. But because of the unwinding, we create tension further upstream here. Topoisomerase. It's what's going to split the DNA, let the tension relieve, reattach. We start at our free 3' end up here at the top of the board. DNA polymerase is going to be involved with copying that strand. And for that one, the helicase moves along, unzipping as it goes. The DNA polymerase follows it, adding a matching base pair, complementary to what it finds on the original strand of DNA. So if this is a C, it's binding a G. If it's an A, now it's going to bind a T. So we get that alternating base pairing to create a brand new double-stranded DNA structure. But we can only go from 5' to 3'. The lagging strand is oriented in the opposite direction. We can't start at the very end because that's the 5' end. That gives us an obstacle. If this is the 3' end, we got to start here somewhere and move in that direction. But the problem is it's not free. We can't start at one end and make our way. So we got to start where we can. We lay down a primer first, and we work our way backwards towards that free end. That is an Okazaki fragment. Now the helicase moves along a little further, creates a little bit more free lagging strand, and we can add down a new fragment. Here right now, another form of DNA polymerase is removing the RNA primer and filling in the gap. Now, when we look at mitosis, that DNA replication was just one portion of that big story. Mitosis is going to be the term given to cellular division of a eukaryotic cell. When you do reproduction, you'll talk about a process called meiosis, where we take your genetic code, essentially cut it in half, so that one sperm carries half of the dad's genetic code, and one egg carries half of mom's genetic code, and when they come together, they give us the proper number of chromosomes we're supposed to have for an individual with a mix of both genetic codes. But here we want the copy of DNA. Here's where that process would have taken place. During a phase called interphase. Interphase is not technically part of mitosis. You'll learn in here and in lab that mitosis has all these different phases with particular landmarks that we observe in each phase. That's what you'll need to be able to recognize, the landmarks, and identify the phase. Interphase is not technically part of mitosis. I want you to think of interphase as a preparatory phase. It gets the cell ready to undergo mitosis. If you want to take a cell and split it to make two cells, there are certain things you are going to need to occur in order to have a hope of pulling that off. One, replication of the organelles, in particular, the centrioles. That's going to occur in the G1 phase, or the G1 stage of interphase. Next, to undergo the S stage. That is DNA replication. That's what we had just gotten done talking about. The enzyme helicase, untwisting the DNA, creating that replication fork. DNA polymerase, copying the strands, so that when all is said and done, you've got two double-stranded DNA molecules. One's going to go to each of those daughter cells. And then lastly, you need that cell to grow. You want to have a cell big enough that when we split the mass, there's going to be enough membrane and enough cytosol to support two new cells. So once you've got a cell that's in a good situation to be able to be split, now you can go through that mitotic splitting. So we had interphase, we make our way through prophase, metaphase, anaphase, telophase, and then we take each of those new daughter cells and go through interphase with each of those. You'll also see a term pop up called cytokinesis, also not a stage of mitosis. Cytokinesis means cellular splitting. So I know you can't see these diagrams very good, but telophase is where you start to see the cell split in half. Still one cell at this point. Cytokinesis is when the split finally takes place and you've got two new young cells. These two new young cells now will undergo interphase, where they replicate their centrioles, replicate their DNA, grow to appropriate size, then they will start to split each. And we go from 1 cell to 2, 2 cells to 4, 4 cells to 8, 8 cells to 16. So here's a bit of a better diagram on what these landmark presentations are going to look like. First, we get the prophase. You are going to see the centrioles. They're not on this diagram, but the centrioles are going to migrate to the poles. The centrioles are going to have spindles that attach onto the genetic material and pull half in each direction. During metaphase is when all that genetic material will line up right along the equator of the cell, with the centrioles on each pole and the spindles attaching to the chromosomal material. So here's the centrioles, here and here at the poles, with spindles attached onto our genetic material. Anaphase is when we get the spindles start to separate them and pull half in one direction and half in the other. So we have our centrioles here reeling in, pulling on the spindles, and literally towing half of your DNA towards one pole, while the other set of spindles pulls the other half of the DNA to the opposite pole, separating that genetic code. So when we do the split, there's no way around having a complete copy of the DNA in each of those new daughter cells. Telophase, again, you can't quite see it on this diagram, but that's where we would start to see the membrane dent in, like it's about to be pinched off. That's the hallmark presentation of telophase. And then finally, once that invagination of the membrane becomes complete, then we have cytokinesis to create two new daughter cells, each with the exact same genetic code as the original cell. Next up is the plasma membrane. And I'll be honest, a lot of the structures we just talked about, we are not going to talk about a whole lot moving forward. We've kind of got that behind us now. Even the mitochondria. What we could have talked about in the mitochondria, we've already have, with our discussions of beta oxidation and Krebs cycle and oxidative phosphorylation. We will be talking about the membrane for quite some time. It is a major topic for where we are right now. Remember, it's essentially the skin that separates what's inside the cell from its external environment. And with a class whose theme is homeostasis, regulation, the integrity of that skin is crucial to controlling what gets into the cell and what gets out. We refer to this structure as a fluid mosaic model. Fluid in the sense that it's got a lot of bendability. You will see that a lot of the structures that make up the membrane are not actually physically attached. They're held together by relative attractive forces. So that creates a very flexible structure, a very fluid structure. Mosaic, because we do see different things on the surface of that cell embedded into the membrane. A mosaic tile countertop is going to have a bunch of different tiles linked together. So when we look at the surface of that cell, we see a lot of different items. There's going to be proteins embedded on there for recognition. Next semester, you'll talk about blood typing. Blood typing relies on the detection of a particular protein on the red blood cell. If we're going to take some cells and make a tissue, we've got to have a way of anchoring those cells together. So you may have proteins on that membrane that are meant to attach to other proteins on neighboring cells. And the one that we'll talk about mostly are going to be transport proteins. Proteins that are specifically involved with moving things across that cell membrane. So here's going to be our fluid mosaic model, all of these different entities embedded in the membrane to give it that mosaic appearance. The fluidity is simply because of this special molecule. It's called a phospholipid. This is the main component of our plasma membrane, the phospholipid. We have a glycerol backbone and an ionic phosphate head. Two long fatty acid tails. Now, we got into a discussion already about basic chemistry, and we got into polar versus nonpolar, ionic and non-ionic. When we got through that topic, I said the main reason we were even getting into that is because I want us to know how these different items dissolve in water. Anything that's polar covalent bonded, or ionic, we're going to consider hydrophilic. Hydrophilic means water-loving. It's going to be dissolvable. It's going to love to be in a watery environment. Things that are non-polar, non-ionic, tend to be more hydrophobic, water-hating. They don't like to mix with water. Fats and oils are hydrophobic. You take some oil, you drop it in water, it doesn't mix well. So here we've got a phospholipid molecule where you've got a very ionic phosphate head and two very long portions of strongly non-polar fatty acid tails. You've literally got a molecule with a dual personality. The top half is going to be very hydrophilic. Top half is going to be very hydrophilic. The bottom half is going to be very hydrophobic. So how is this molecule going to behave if we put it in a watery environment? The body is one big watery environment, right? When we take a phospholipid, put it into a flask of water and shake it up, we see the phospholipid take on a very particular arrangement to make itself happy inside that solution. This is called a mycele. And look at what's happening here. The blue is representative of our hydrophilic head, phosphate head. The red strands are representative of our fatty acid tails. Look at this little ingenious way it makes itself happy in a watery environment. It forms this sphere. And it takes all of its hydrophobic fatty acid tails and points it internally. So this is in a watery environment. So water, 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 water. The only parts of this mycele that has to interact with the water directly is all the phosphate heads. And they've got no problem with that because they love to be in water. Meanwhile, the water-hating portions of the phospholipid are oriented internally, and they've sequestered themselves off so they don't have to interact with the water. They're using their own phosphate heads as a shield to keep the water away. So they take on this orientation naturally, unbounded. These are not bound together. They're held in this position because of the relative attractive and repulsive forces the different pieces of that phospholipid has for the water in which it sits. Simple attractive and repulsive forces create this mycel structure. Now, we don't have much of a use for mycel. This is not in any way, shape, or form our plasma membrane. But the plasma membrane does use those same relative attractive and repulsive forces to make its particular structure. What we get is called a phospholipid bilayer, and you saw that, whether you realize it or not, on our previous diagram. Phospholipids, those red dots, and rather than create a sphere with the fatty acid tails pointing inside, we get this double layer, but it serves the exact same function. You have fluid inside the cell, water inside the cell. You have water outside the cell. You got a layer of phospholipid or hydrophilic heads facing internally. You've got a layer of hydrophilic heads facing externally. So just like the mycel, the only parts of the phospholipid that has to interact directly with the water is the heads. And we point the fatty acid tails internally like the meat of the sandwich. And once in there, just like our mycel, we can avoid them ever being in contact with the watery environment. Phosphate heads, hydrophilic, which is great because we're going to have water here internally, water here externally, and this sequestered off hydrophobic region now in which the fatty acid tails are perfectly happy. This is a very stable structure. But remember, fluid mosaic model. These phospholipids are not physically attached. They're held there because of the relative attractive and repulsive forces that the phospholipid has with the water in which it sits. So we could take this phospholipid, that phosphate head, and attach a label to it. Leave it alone and come back, go to lunch, come back and look at it 45 or so minutes later, and now that phospholipid may be over here. Remember, there's nothing binding it in place. It can migrate along the surface of that membrane. An analogy I use a lot to look at the surface of a cell is if we had, picture a big fish tank. Half fill it with water and dump ping pong balls into that tank. Just enough so that those floating ping pong balls will cover the surface. We look down on those ping pong balls and you see the phosphate heads. Nothing is stopping this phosphate head and the tails that are pointing inward from migrating its way through that membrane, not much different than someone would migrate through the crowd at a concert. But where we're taking this structure is we're introducing the membrane in such a way that we're going to be working on how do we get things in and out of the cell. Well, before we get there, we already said that we can move the phospholipids around the surface on either side. Can we ever see easily this phospholipid here with its tails flip and become part of the other side of the membrane? The answer to that, if it's possible, is that not likely, and certainly not easily. Look what's being asked in order to take this phospholipid and orient it now so that it's on this half of the membrane. Here is your watery environment. Here is your watery environment. The phosphate heads are very hydrophilic. They love water. What you've done to keep the fatty acid tails happy is you've created a very strong hydrophobic environment. In order to get a phospholipid to go from one end to the other, you're asking a very hydrophilic portion to work its way through this very strong hydrophobic environment all the way to the other side. Looking at this as a diagram, you're like, well, that's not that hard. But this is not really a scale that we're familiar with. Picture if we're looking at a football field, with the end zones being our phosphate heads, and from goal line to goal line being our hydrophobic region. And you had one person in one end zone wanting to get to the other end zone, and every step of the way they're going to get repelled by a football player. These phosphate heads, even though they're anchored onto the fatty acid tails, they're repelled outwards by their own hydrophobic environment. We will not be able to have something that's very hydrophilic make its way through that strong hydrophobic environment to make its way to the other side. And that's critical for controlling what gets through that particular membrane. That entire concept there makes the membrane a very effective barrier to anything crossing, coming in or going out. It sets it up in such a way that we gain control over what enters and what leaves the cell because of the structure of that phospholipid bilayer. So again, when we talk about transport now, remember, this was the cell. Here was all the organelles we just talked about. Here's our plasma membrane, phospholipid bilayer. When we talk about membrane transport now, we are talking about things going into and out of the cell. Exchange between the intracellular fluid, or the cytosol, with the interstitial fluid that's found extracellularly. Some of the concepts we'll talk about with regard to transport, we will use again in AMP2, but not into and out of the cell. We may talk about things going from the blood into the interstitial fluid. So some of the concepts will remain the same, and we'll add on to those to make them applicable once we get to that topic. But anything we're talking about right now in regards to transport is into and out of the cell only, and disregarding any other movement. So when we look at the phospholipid bilayer and we start thinking about how it controls different items getting into and out of the cell, I described this structure as ping pong balls earlier with good reason. I didn't pick something shaped like a cube that floats. I picked a round ping pong ball. When you put ping pong balls in that water, they don't pack tightly. There's always going to be a little space in between the ping pong balls. So right off the bat, if we want something to get through that membrane, the size of the molecule comes into play. If it's a small molecule, it can squeeze in between the holes. If it's a big one, it cannot. But not everything that's small is simply able to scoot through the holes and get into and out of the cell. Because remember, as soon as we get through the holes, now we've got a very strong hydrophobic environment. So something small like an ion that carries a charge, that's very hydrophilic. It could easily fit in the holes. This would be like throwing a golf ball into a garage door. But once it gets through the door, it immediately finds itself in this very strong hydrophobic environment, and it's immediately repelled away. The structure of the membrane dictates what can get through naturally. In order for something to pass directly through the membrane, it has to be small enough to fit between the phospholipid heads, and it has to be hydrophobic enough to be at home in the hydrophobic environment created by the fatty acid tails. Only by checking both of those boxes can we allow things to move directly through the membrane, seemingly unchecked. So something like oxygen and carbon dioxide drift right through. Small enough, non-polar. Perfect. But if it's too big, or if it's too hydrophilic, like an ion or a polar molecule, it needs other assistance to get across that membrane. And when we say assistance, protein transporters, you will see diagrams similar to this as we go. Sometimes they'll be very basic, like a protein tube, creating a larger hole to accommodate the larger molecules. But also, what about if this was an ion? Very small, but very hydrophilic. If that's an ion, it does not want to interact with the fatty acid tails because they're very hydrophobic. By putting a tube in there, it allows them to pass through without their opposing characteristics repelling them. Sometimes you'll see them structured like these Pac-Man looking shapes. Something comes in, binds, and then it opens up to the other side of the membrane. Some of those can be very specific for one particular type of solute. Other times they can be very general, letting anything pass through. So as far as this goes, you need to know the basic cellular structure. You'll get a couple questions on those. But more importantly, you need to understand the plasma membrane. This is going to be one of those topics that will haunt you down the road. If you don't understand the structure of the membrane, it's going to be that much more difficult to understand the transport section. Likewise, if you don't understand the transport section, you're going to have a very difficult time understanding the neurophysiology that we'll be eventually getting into. You should understand the stages of protein synthesis, transcription, translation, and you should understand cellular replication, which involves DNA replication and the process of mitosis. So we're going to pick it up. The very next section we'll talk about, we're going to jump right back into that plasma membrane again, reorient ourselves with it, and then look at specifically how we get certain entities from one side of that membrane to the other in a controlled manner. Thank you.